The graph shows the various measurements taken from the metaphase of the mitosis onward. The graphs are to scale when to compare the one another. So in this graph, we are supposed to identify the different part of mitosis with the reference of the graph values. For example, the graph X, Y, and Z, they represent the distance of these components after metaphase. So we can see these different graphs and their representation. For example, the graph X, it represents the distance was at a certain point, and after metaphase, this distance decreased to 0. For example, when we see these 2 points, like distance between the chromatid and the pole was at a certain point, but after metaphase, this distance decreased to 0. So we can say that that's a point, the distance of centromere from the pole of the spindle. In the same way, we shall move to the second point here. And in this second point, Y graph represent that the distance was maximum and then it surged to increase a bit. So we can see that the distance was maximum between 2 points, and then this distance increased a bit. So we can say like the distance between the pole, it was maximum, and then this distance after telophase to the cytokinesis. So when these poles move a bit apart during cytokinesis, so this distance surge up, and in this way, the distance between the pole of the spindle. While the Z represent, that's another, the distance, first the distance was 0, and then after metaphase, this distance increased to the maximum levels. For example, the distance between these two chromatid, before it was 0, but after metaphase, during anaphase, they move apart from each other. And when they're moving apart from each other, so this distance is increasing to maximum. So on the basis of this idea, we got idea, the distance between the sister chromatid, that is Z. So here, the distance of the centromere from the pole of the spindle is represented by the X graph, while the distance between the pole of the spindle is the Y, and the distance between sister chromatid is Z. So here, that's the original questions. We're going to move toward the original question. So the answer will be R, the C. So the X, that's going to represent the distance of centromere from the pole of the spindle. So this distance was at a certain point when after the metaphase, when these two chromatid move apart from each other. So this distance, reach to the pole, these two chromatid, they move apart and move toward the pole, and this distance become 0. While in the Z, when we see that the distance between the pole of the spindle, it was maximum between. And during cytokinesis, it increases. And until the end of the mitosis, it remains same, but during cytokinesis, it increases a bit. While in the Z, the distance between sister chromatid, first it was the 0 in prophase, metaphase, but during anaphase, these two chromatid will move away from each other, and this distance will increase. In a similar way, we can say another type of questions, all the data is similar. For example, the X represent, so that is the distance of centromere from the pole of the spindle, while the Y represent, this distance always remains same during the mitosis. Here, we are not entering into the cytokinesis. So in this way, the distance between the pole will remain same. What about in Z? The distance between the sister chromatid, first it was the 0, and then it surged, increased to the maximum. So that was the idea how we can identify the answer of these questions. Hope you got idea. So thank you very much for watching.